So John has been working in the biopharmaceutical development industry for over 30 years, working for international companies such as MSD and Fujifilm. And he is now working as the Chief Technologist at CPI, who work with industry and academia to support development and new therapeutics and their manufacturing processes. So one area that indeed the vaccine industry and the CPI have been looking at into recently is the mRNA vaccine manufacturing, because of the potential for high potency, prospects for rapid development and deployment, and compact manufacturing. And it's this topic that John has kindly offered to present to the Vax-Hub today. So we'll say thank you to and welcome to John, and I'll hand over to you. Thank you.
Yes, thank you, Andrew. Just get the slides up. Oh yes. So hopefully you are seeing a presentation on your screens.
Yeah, we've got that. Thanks, John.
That's great. Okay. So hi, as Andrew previously advised, I'm John Liddell, and I'm Chief Technologist at CPI's Darlington facility. I'm going to talk today about RNA vaccine manufacture, and the graphic shows a very important part of that, which is the T7 polymerase that has been used to make RNA, and we'll come on to describe a little bit more about that. It's a very interesting enzyme. So the outline of the presentation is as shown here. We've got give you a little bit of background to CPI and CPI Biologics. It's worth spending a little bit of time on vaccine background in terms of types and COVID-19 vaccine in development, because that's kind of an ongoing thing. Then cover a dive into mRNA technologies, including what we would have in inverted commas conventional, and then the self-amplifying mRNA vaccines. And given my experience has been largely with the self-amplifying, a lot of the presentation is dwells on self-amplifying RNAs. Then conclusion and future prospects, and then on to questions afterwards. So first, a little bit of background to CPI. We're part of the High Value Manufacturing Catapult, which is supporting the development of advanced manufacturing in the UK. Our centres are broadly, with one exception, in the northeast of England. So just to remind you where things are, we've got ourselves in Darlington. We have an industrial biotechnology group at Wilton looking at, I guess, more industrial biotech applications, industrial enzymes, making fish food from natural gas. Our centre at Sedgefield are involved in formulations and printable electronics, and we have a small-scale printable electronics facility in Newton Aycliffe. We're in the process of establishing a medicines manufacturing innovation centre near Glasgow, which will be looking at improving the manufacture of small molecule pharmaceuticals, so looking at continuous manufacturing and applications of PAT. In terms of biologics, the centre in Darlington is just about exactly 5 years old now. It was a £38 million state-of-the-art facility, has 5,000 square metres of lab space, and is equipped for microbial and mammalian expression work. We've worked on monoclonal antibodies, we've dedicated suites for working on viral vectors, we've worked on microbial therapeutics, VLPs, antibody drug conjugates, and nanomedicines, which would include mRNA. 13 flexible labs which are equipped to BSL 1 and 2, and we have some suites that are being modified and prepped up for being able to do GMP manufacture. And at the moment it's ISO 9001 certified, but clearly it will be, some parts of the facility will be operating as GMP in the very near future. In terms of the capabilities of the group in Darlington, well, in terms of upstream, we can cover things from initial screening in multi-well plates or flasks, moving on to process optimization using multi-parallel bioreactors, things like Amber 15s or Amber 250 systems. And these give you a lot, because you're looking at a large design space, you can do a lot of optimization very rapidly. The usual approach would be then to confirm the optimization by scaling up to 10-liter bioreactors, and depending on the product requirements, up to, say, 50 or 200-liter bioreactors, which are all of single-use format. Matching downstream development capability, it's going from, again, the application or exploration of large design spaces using slurry plates or robocolumns to get a feel for media capacities, media screening, and then doing something more chromatographic on a reduced set of conditions using robocolumns on liquid handlers. Again, bench scale confirmation, and potentially taking a developed stage 1 or stage N process to be able to make material to then look at stage N+1 that's going back to small scale. And we have pilot scale equipment that will go up to now active process levels, probably about 200 liters per hour flow rates. A range of self-packing columns based around the Axichrome GE format. Analytical capabilities very broad to support the broad range of products we're supporting. So everything from titer, purity and identity through to host cell protein measurement, endotoxin, host cell DNA, product aggregation through various chromatographic methods or dynamic light scattering, microflow imaging, nanoparticle tracking. We have high resolution capability, so we have 2 mass spec systems, one with hydrogen deuterium exchange for understanding modifications to secondary and tertiary structure, and an advanced CD system for looking at CD changes that have been seen in proteins. And we have been using that actually with the mRNA project. It's a very informative technique for looking at mRNA structures. Product formulation ability testing, and we have next generation sequencing capability for which we've used for looking at mispackaging of host cell genomes into viral vectors, for example. We have quite a comprehensive suite of formulation capabilities. So classical ones like liquid formulation development, we have stability analyzers and stability incubators for doing informal studies. We have lyophilization capability, you can look at collapse temperatures, cycle development and small scale lyophilization. But maybe more unusually, in collaboration with another parts of CPI, our formulations group, which they have equipment for producing nanoparticle formulations. They have some precision nanosystem microfluidic devices, and that will provide a scalable solution to make micro and nanoparticles. And that's quite important for mRNA production, as we'll come on to. Moving on from that background, just say something about vaccines and what vaccines are really all about. In a very simplistic term, a vaccine primes the immune system to recognize an antigen, which allows the immune system to respond rapidly to subsequent challenge by the antigen, i.e. a pathogenic organism. And classically, there's been two fundamental ways. The original and favorite ways have been to provide the antigen in its final form to the patient. And you've got several ways of doing that. You've got classical vaccines, which would have used or still use live or live attenuated or non-viable cells. And these are vaccines you will have experienced if you've had mumps, measles, or rubella vaccines, polio vaccines, all in that classical vaccine format. Conjugate vaccines, where you've conjugated a glycan to an immunogenic protein like the crippled diphtheria toxin CRIM197, things like Prevnar and a similar Merck product have been developed for pneumococcal diseases. Also things like whooping cough. If you've had whooping cough vaccine, you will have had a conjugate vaccine delivered to you. VLPs, these are clearly capsid proteins that are formed of virus-like particle, but do not contain any genetic material. And vaccines that are out there that have been as VLPs, if you've had the HPV vaccine, that's a VLP. Other ones might be more obscure, like for hepatitis B. And these are produced by a wide range of systems that people have used. Picopastoris, Saccharomyces cerevisiae, baculovirus, and then coming on to classical subunit vaccines, where the actual vaccine is a subunit protein produced by recombinant technology in whatever system you fancy, typically either E. coli or mammalian cells, or maybe baculovirus, and that could be as an FC conjugate or conjugated antigenic species, or associated with an adjuvant. And until quite recently, these have been the main form. So you delivered the antigen to the patient in the final form. But if you then look at the graphic on the right-hand side, there's various steps on the way to making an antigen. The antigen is after DNA has been translated into RNA and then transcribed into protein. You can go further up the chain, so you can supply the antigen in a form where the body will produce the antigen itself, and that be as a DNA vaccine, which either be as a plasmid or using viral gene therapy, where you have using adenovirus to deliver the genetic the genome to the cells that you want to allow the cells to make the antigen themselves. And then more recently, mRNA vaccines, where you've skipped a step, you've translated the DNA into an mRNA, and you deliver that to the cells. You've only got one membrane to cross before you can the cells will express the antigen. And this is I guess the most recent, probably within the last 10, 15 years for the bulk of work on this. And these need to be formed into liposomes or polyplexes to be effective. And that's probably the main sort of learning point that had, among the many learning points, that has allowed people to develop mRNA vaccines. In terms of vaccines, pros and cons, I mean, some of these things are pretty obvious. The classical vaccines require specialized high containment. The organisms are all different. Conjugate vaccines, they're because you've got glycans and immunogenic proteins, and conjugation manufacture is reasonably complex, and typically are targeting bacterial diseases, things like mycobacteria, which have a glycan coat. Virus-like particles, again, it's the complex characterization and manufacturing controls for consistency. Subunit vaccines, well, again, they cover a full range. They're difficult to platform because all the subunit types are going to be different. Viral vector vaccines, you start to get into things that are more platformable. If it's a viral vector, probably the same viral vector can be used for umpteen different types of DNA transgene. If you're delivering a plasmid, or producing a plasmid, probably the process to make that will be independent of the plasmid you're making. On the viral vector vaccines, there's not been a huge amount of past experience. The first one approved for human use was only licensed back last December for Ebola. And for the DNA and RNA vaccines, although there's been lots of animal data, to date there's none been licensed for use as a human vaccine. But tremendous potential, as we'll come on to. Just to indicate where people have been focusing their efforts, the WHO produces a list of COVID-19 vaccines in clinical trials around about on a monthly basis towards the end of each month. So this is the data from August 2020. And looks like it was about 20th of August. At that stage, there was a total of 30 different COVID-19 vaccines in development or in clinical trials, and there's a total of almost 170 in development. And you can see the types of vaccines that were being considered for in clinical trials. 20% of these are RNA vaccines. 13% are DNA vaccines. So that's almost a third of the total are based on a new vaccine approach. There's clearly still a lot of interest in inactivated vaccines and subunit vaccines. But concentrating on the mRNA vaccines, why are people interested in these? Well, for a start, mRNA is non-infectious and it's a non-integrating platform. There's no potential risk of infection or insertion of mutagenesis. mRNA is degraded by normal cellular processes, which can be an advantage and disadvantage. In vivo half-life can be regulated through the use of various modifications in delivery methods that we'll come on to. It's a minimal genetic vector, so anti-vector immunity is avoided. It's very potent, small doses are required for protection. For the self-amplifying RNA, the dose levels that people are looking at for a vaccine are in the order of 1 to 5 micrograms per dose. It's a little bit higher for non-amplifying mRNA vaccines, maybe about 25 to 30 micrograms. So coming to the processes are typically quite compact. It's quite an efficient process. In vitro transcription reactions are very efficient. And because the process is probably going to be independent of the size of the antigen or the type of the antigen being expressed, it's mRNA you're making, it's probably a platformable process. So you've got this small process. You're not doing things in 1,000-litre tanks or having whole tank farms of 1,000-litre tanks. You've got a small, compact process. So what's the deal with mRNA vaccines? Well, there's two basic approaches that we've covered. There's what I've called conventional non-amplifying mRNA. And for COVID-19, there's a number of these in development. Moderna's 1273 vaccine is based on non-amplifying mRNA. The CureVac vaccine as well, the Pfizer CureVac vaccine. And the structure is the mRNA is as indicated here. There's and this is as a result of years or many years of understanding things that control the stability of mRNA. So you have a poly A tail, maybe 30 or 40 adenosines at the end of the 3' end of the mRNA. You have the transgene to make the antigen, a capping reagent at the start of the sequence, which we'll come on to. This is quite important for effective expression in eukaryotes. If you don't have the capping, it's the mRNA tends to be degraded because it's not recognized as being wanted for translation. Untranslated regions before and after the transgene, which indicates what the transgene is that's going to be expressed. And then this cellular machinery just goes off and makes the antigen, which in COVID-19 vaccines is typically the viral spike protein. The overall size of these things is probably around about, well, in the COVID-19 vaccine, the spike proteins around about 4,000 base pairs, or nucleotides rather. So the overall size of an mRNA vaccine is something under 5,000 nucleotides. Self-amplifying mRNA uses a slightly different approach. And this is why you can get away with a smaller dose level. You've got the same feature as the capping, the cap of the mRNA, the untranslated regions before and after the transgene, but ahead of the transgene you've got 4 sequences that have been taken from an alpha virus. And these are 4 proteins that together make an RNA-dependent RNA polymerase. So they will be expressed, they'll make this replicase, which then will take the transgene and make multiple copies of the, multiple copies of the RNA, and then multiple copies of which will then be expressed when it comes to express the transgene, making copies of the antigen. So you just need smaller amounts of self-amplifying RNA because it just makes more of itself through the production of the replicase. In all cases for so just before going to that, this increases the size of the mRNA quite significantly. The replicase is probably around about 10 kilobases in size, so it means the overall size of COVID-19 self-replicating mRNA vaccine is probably about 15,000 nucleotides. In both cases, though, to be able to get the mRNA delivered to the cells, production of a lipid nanoparticle is the typical approach that's been formed. And this has been key to being able to deliver mRNA in an active form to the cells. I'll say a little bit more about these. It's quite a complex lipid mixture and requires complex some quite complex processing to achieve. So having described mRNA, how do you make it? Well, it's we show here, this is some of our small scale experiments. This is clearly an Amber 15 Tic Tac boxes, but it's made by one-pot enzymic synthesis. And you take a DNA template, which would typically be a linearized plasmid, but we have also, people have also been using alternate templates such as Touchlight's docubone DNA, which you take the ends off and effectively then have a linearized DNA sequence. And the advantage over that compared to a plasmid, if you want to make a plasmid, you usually have a cell line that makes a plasmid. And some of these big mRNAs require a big plasmid, and that large plasmid may be expressed in small levels. Also in the pot, as well as your DNA template, you'll throw in a T7 polymerase, which is clearly going to do the transcription of the DNA into mRNA. A pyrophosphatase, because the reaction generates pyrophosphate, And if you don't know the pyrophosphatase there, you'll just generate a lot of insoluble pyrophosphate that's going to cause you problems later on in processing. It's one of the RNAs are one of the bugbears of working on mRNA. So you want to have everything swimming in an RNA inhibitor, an RNase inhibitor. You'll also have a set of equal amounts of all the 4 nucleotides in the mix and an RNA capping reagent. For the synthesis, you'll throw in a DNase to chew up the template. And if you're doing the reaction at around about 37 degrees, in 2 hours, you'll have around about 5 grams per liter of mRNA. So it's very, very high yielding. I'll just see if I can get this animation to work. Yep, that's fine. So the key to all this is the T7 RNA polymerase. It's the smallest RNA polymerase. It's just under 100 kilodaltons in size. It's one of the simplest RNA polymerases out there. It requires no accessory proteins or cofactors, and the crystal structure was produced about 20 years ago with from Tom Steitz's lab with a DNA template bound to it. And the X-ray map will show you the transcription taking place. It's a very interesting little enzyme. It's doing several things, and it's only driven by the triphosphate to monophosphate reaction, generating the pyrophosphate as we mentioned. So it's doing all this, and it's doing several things in order to function. It needs to bind to the promoter region of the DNA strand. It then separates the DNA strands to free up the 3' chain for RNA synthesis. So generating this transcription bubble that's around about 15 bases. It then needs to move the template strand past the active site, incorporating a capping molecule at the start. It then polymerases RNA nucleotides complementary to the template strand, and then detaches the strand from the template at the end of transcription. And then it starts again with another DNA strand. And it's doing all this through mechanisms that are still under argument after about 20 years of having the structure. The favourite at the moment, it's a Brownian ratchet. So the action of binding the nucleotide moves everything forward one position, and it can't go backwards, and that's just driven by Brownian motion. The reaction, it's from molecular modelling, the reckoning that it takes, molecular mechanics calculation, takes around about 20 microseconds per base pair. So with the number of bases it's incorporating, it probably takes about 4 minutes to move down a DNA strand to generate one mRNA molecule. We mentioned capping, and this is a very important little piece. The capping reagent is shown here. And the need to cap, as I said, is because a cap1 structure is shown here, i.e. you've got a methyl group here, and this 3-to-3 triphosphate linkage to a nucleotide is for what they call cap1. It's found in all higher eukaryotic mRNAs, and it's really necessary to ensure that your mRNA is then expressed suitably in the eukaryotic cell. This is a reagent that has been developed by CleanCap. So CleanCap has been developed by TriLink Biotechnologies. You just throw it into the mix, and you get 99% of your mRNA is capped. It doesn't require any other separation or other enzymes. There's no need for purification. There's no need for specialized capping enzymes, but it is expensive and it is proprietary. And probably about 60% of the raw material cost of making mRNA is tied up in the capping reagent. There are some alternatives to clean cap that have been, but they're also proprietary, and they're probably also equally expensive. But this is a very effective if you can bear the cost of being able to make mRNA on a large scale. For saRNA purification, the method we've been using is to make use of the size of the molecule. Let's say it's around about just under 12,000 nucleotides. So it's actually a structure of 4 million Daltons. It's around about 50 nanometers in size, and that size varies somewhat with ionic strength. It's a unique purification situation. You know all the impurities that are present. You know what the residual enzymes are. You know the polymerase. You know the features of the DNAs, you know, the pyrophosphatase and the RNase inhibitor. Other things you'll have there are residual nucleotides and excess capping reagent, and DNA fragments from the hydrolysis of your DNA template. So the typical approach has been to make use of that the size properties, and to do a sort of almost a process that leaves the mRNA as the remaining product in solution. So from the reaction, you'd typically do a tangential flow filtration, where you would be washing out small species like the residual nucleotides, DNA fragments, some of the smaller enzymes as well, if you choose your membrane size right. Then, because your binding capacity if you want to bind and elute from a chromatographic media, is probably going to be quite low. You do a negative capture step, so you're having a flow-through step that will bind any residual proteins and any residual small molecules. Then onto a second TFF step where you do buffer exchange to get you into the correct buffers to be able to do encapsulation, and the final TFF step to adjust concentrations and formulation buffers before doing sterile filtration into the final product vials, or the final bulk drug substance or drug product before vialing. So it's a process that makes use of the large size of the mRNA. And then we come on to encapsulation of the product. That is key to being able to get an efficacious product. Typically for these RNA products, and I think this has been learning from making DNA as a therapeutic as well, use a 4-lipid system, which would be a cationic lipid. And again, these are typically proprietary. Cholesterol, which is give membrane structure, third lipid whose name I cannot remember anymore, and a pegylated lipid I guess prevent the nanoparticles, well, keep the liposomes and lipid nanoparticles separated. And say we have been using these precision nanosystem devices, microfluidic devices, to make lipid nanoparticles using these formulations, which would be typically in this sort of ratio, quite defined ratios. And this is really quite an important thing. This is a gel showing, this is the mRNA here. This is mRNA without encapsulation, mRNA with encapsulation. This is if you've left the mRNA with some RNA, so you see just get complete degradation to a fuzzy smear at the bottom of the gel. If you have encapsulated your mRNA, even when being incubated with RNAs, you see no degradation at all. So it's really quite important to get the molecule in a position where you can actually deliver it to patients. Dose levels are quite, I say, actual doses are small, but also concentration is quite small. And at the moment, storage stability is being defined as -80 storage. In terms of some conclusions, it's well, RNA potentially a very flexible and compact and platformal gene delivery technology. People have looked at it as well for in situ delivery of antibodies. So rather than having an antigen, you make an antibody or provide the sequences for making the antibody in the mRNA. It's well suited to a rapid vaccine response. Again, it's a compact process. You could see as well how, because of the very small dose levels, and with advances in technology, you could even see how you could get to having, it's almost something you could envisage having a shoebox that would make some mRNA for any vaccine. All you require is a template, and that template could be, I say, a plasmid. The template could be a non-plasmid based template. Its production of these is well matched to CPI's capabilities and expertise. We've had a lot of experience with cell-free expression, which has been very helpful in development of the synthesis of the mRNA, and we've had a lot of experience with microfluidic nanoparticle formation, and again, that's been very important for this this project. mRNA is typically an unexplored technical landscape. There's significant scope for innovation, both in terms of understanding how to take the reactions forward. The reaction is at the moment quite efficient. I would say that things that would merit further work around about the capping reagent, that's a very high cost, formulation and storage, and some of the lipid nanoparticle manufacture is a little bit the lipids are typically only solvent soluble, so you might have the lipids dissolved in ethanol and then carry out a precipitation effectively of the mRNA into the liposome through dilution. So having formulation methods that would make achieve the same sort of effect, but avoiding the use of solvents and dilutions would make the process even more compact and simple to operate. Just some acknowledgements. We've clearly been working with Robin Shattuck's group at Imperial College on the mRNA work. They've been progressing. As an example of how fast these things can be developed for mRNA, I think Professor Shattuck received the sequence for the COVID spike protein round about the end of January this year. That was able to be worked up into a plasmid and provided to a CRO to manufactured phase 1 and phase 2 material back in May, June. These then went into phase 1, phase 2 clinical trials, which will be giving readouts in November time. So it just shows a very rapid approach to a pandemic that can be achieved with these sorts of technologies. And that's from a standing start. If you were to do it again, we'd know a little bit more and hence some of the things to direct things at. And the other acknowledgement is clear to the CPI saRNA vaccine team, who have been working across platforms at CPI with the biologics and formulation platform. Just a couple more slides just to say that we don't just work on saRNA vaccines. We've been working with the Future Vaccine Manufacturing Research Hub. This is the one being run out of Imperial College. So we've been working on VLPs for polio with Leeds University. A new multigenic vaccine for pneumococcal diseases with MBio, and be looking at some of the formulation challenges that have been associated with this sort of quite complex subunit vaccine, and with Exivian for an antigen candidate for Zika, which has got quite some complex oligomeric structures and some quite complex glycan formations, which we're helping Exivian characterize. My final slide was just a little bit of a refresher on COVID-19 vaccines secured for the UK. I have to say that all these are still going through their clinical trials and nothing has been approved as yet, and in terms of efficacy as well to be demonstrated. So we've got the virally vectored vaccines, of which we've got the Oxford AZ vaccine, a similar vaccine from Janssen. Subunit vaccines, we've got the Novavax vaccine made in insect cells, which Fujifilm are in the position to manufacture in the UK, as well as their sites in the US. The GSK Sanofi vaccine, another subunit one using the subunit, I think has come from Sanofi, the adjuvant has come from GSK. mRNA vaccines, the saRNA is Imperial College that we've just discussed. The other one is more conventional mRNA vaccine from BioNTech and Pfizer, and the whole virus vaccine, which is coming from Valneva, who have a manufacturing facility in Livingston, Scotland, and that starts phase 1 trials December this year. And Her Majesty's Government has invested in the facility on behalf of us all, which is encouraging. So I think watch this space. A lot of these are going to be giving readouts, apart from the Valneva one, probably around about the end of this year. So we'll see see which one's going to be the winner, which one's going to be first one out of the box. And that was the presentation. So thank you for your attention. Happy to take questions now.
Thanks, John. That was very interesting and clearly very pertinent. So for the taking of questions, so as I said at the start, you can either raise your hand and I'll come to you, or if you post a question in the chat and we'll go through those. So I saw quite a number of questions going on in the chat, but also Nestola, you had your hand up quite early on in the presentation. So if we come to you and if you want to ask a question, then we'll go to the chat questions.
Yeah, just for one question from my side that I also posted in the chat. When we are talking about the reaction of the mRNA, which are the critical process parameters that we need to watch at? So is this a reaction which is straightforward, or you need a lot of fine tuning in terms of pH, agitation, oxygen that you need to fine tune properly?
I have to say we've been making a lot of use of Robin Shattuck's group's understanding of the reaction. So they've got developed quite and have run some DOEs on some of the important parameters. I'd never say that optimization is ever complete. I think there's still some things to go at there. There's a lot of things that it will certainly make around about 5 grams of RNA per liter, but there's a lot of excess clean cap left over. You tend to have clean cap present in excess, and given that's quite an expensive reagent, it might be useful to see if you can get away with lower levels or different feeding strategies that might enable you to get away with less of that. Temperature is a good one as well. People always just opt for 37 degrees. Could there be some advantage in terms of product quality, maybe by running slower? It's a T7 polymerase. It's I guess it's it'd be used to seeing working in E. coli, and E. coli is quite a broad temperature window, so there's probably some things I would suggest in terms of temperature. You might want ultimate productivity, you might want to also a little think about quality. Is that okay?
Yeah, thank you.
So I'll do a couple of questions from the chat, and then we'll come back to you, Barry. So Dan is asking, are you attempting to get one mRNA molecule per liposome?
Well, that's a very interesting question. I think given the size of the liposomes and the size of the molecule, I think it is effectively one mRNA per liposome, although it's not something we've actually attempted to control. The liposomes are, when we've measured them, aren't that much larger than the mRNA itself. They're around about 70 nanometers. And they will go through a 0.2 micron filter. So we think there's probably, on average, one mRNA per liposome. There's a little bit of controversy as to whether all the mRNAs within the liposome, have you got any mRNA sticking out that might then bind to other mRNAs, giving aggregation. That's why I say this is a little bit of an unexplored landscape. There's lots of things to be further understood. The technology clearly works. People can get immunogenic response. You get you get good stability of the mRNA in vivo, but I'm sure there's multiple other things to be worked on.
Okay, and then so Steve has asked, being so dependent on proprietary reagents, can you give an idea of the sort of cost per dose for single saRNA vaccine? What sort of timescale does a production run take? Presumably fast compared to fermentation-based systems. Well, yeah, I guess.
Yeah. Yeah, the one about proprietary reagents is clearly quite a pertinent one. You're dependent on a sole supplier for a number of these things: the clean cap reagent and the lipids. To an extent, it's probably not that different from other processes as well. I mean, if we always used to have these arguments about, well, you're depending on GE for making your chromatographic media, and clearly you could go off and redevelop your process around about different media from a different vendor, but it just take you so long, and so you're kind of stuck with GE. There's clearly be some supply chain risks where you might want to stockpile material if the if shelf life is sufficient. And again, I think this is where it's an unexplored landscape. There are other capping reagents or other lipids. How much work would it be to change things over? And in terms of production timescales, well, it depends how fast you want to run. If you're operating, say, just during a day, you can probably make a batch of mRNA in 2 days. So it's really fast. Mm-hmm. You might have a third day to then do the encapsulation, but it's compared to typically, and it's always difficult to, if you are working 24/7, you'll do things faster, but typically MAB processes might be about 5 days. Here you've got a 3 or 4 day vaccine process, so 3 days.
Yeah. Barry, we'll come to you.
Hi, yeah, this is great work. Thanks. One of the practical issues for widespread distribution is the temperature stability. So what are the prospects for getting a more temperature stable version of this, and what kind of approaches are being taken?
Yeah, I think it's the, again, it's because things have happened quite fast. And I think people just haven't quite done the work. There There are other lipid nanoparticle systems on the market, and these mRNAs have typically been making use of technology that was developed originally for DNA vaccines and for saRNA. So things like patirisan, it uses a lipid formulation, although it's only a small RNA, it's around 30 bases. It uses the same lipid formulation, and there they've got stability data going out for about 2 years, and that's at, I think, it's either minus 20 or 2 to 8. And I think it's some of I think there's lots of things to be done around about some of the excipients you might have present. The current excipients present in the nanoparticle formulation are quite basic. That's not so much in terms of charge, but they're very, very simple. I think more advanced formulation components using some best practice from other formulation systems should yield benefit. I think there's really a good prospect with a little more work being done to get a more stable nanoparticle formulation.
Okay, going back to the chat. So Thomas is asking, is there a chance of RNA binding to the outside of the liposome? Is it possible to characterize it or prevent it?
Yeah, I'm sure there is some loss of RNA to the outs that just doesn't get encapsulated. From measurements made, it looks like the encapsulation process is probably around about 95–97% effective. So you don't detect a lot of mRNA that's not been encapsulated in a liposome. Again, another question is, you've got 4 lipids there. Have all the liposomes got the same composition? What sort of lipid interchanges are between liposomes? I'm not quite sure. There's people have done TEMs and sectioning, and you can see the structure of the liposome. It would seem to be potentially possible, but in terms of what we've measured, it doesn't seem to be a huge amount bound to or not incorporated in the lipid nanoparticle. Yeah.
Okay. So from Dan, is sterile filtration challenging for liposomes?
The size of the nanoparticles means that they can be 0.2 micron filtered, which is the big advantage. The size of the nanoparticles is just under 100 nanometers. Typically, there's a bit of a size range, and they will flow through a 0.2 micron filter. So they can be terminally sterilized through a 0.2 micron filter, which it would be a bit tedious if that wasn't possible. There have been reports of the potential for nanoparticles, lipid nanoparticles, to aggregate, and that's again tied up with the formulation and possibly also the mRNA sticking out into the surrounding media, which gives rise to binding to another similar nanoparticle and leading to some sort of aggregation that can mean that material would then be held up on the filter. Mm. But I think with a suitably designed encapsulation process and preparation for the 0.2 micron filtration using excipients and also the concentration of the lipid nanoparticles, at the moment that's not a huge issue, but certainly one to watch.
Mm. Slightly relating to that, so Andrea is asking how stable are liposomes in terms of when you're doing diafiltration, when the buffers are changing, the solvent environment may impact these particles?
Yes to all these. It is you might be transiting through the PI. Watch how you're doing that. You want to watch your concentration. You want to watch the sort of pump you're using. So peristaltic pumps where you get compression of lipid nanoparticles as the rollers go around. Don't do that. Use a different sort of pump, maybe a quattro pump or even a displacement pumps. There's the it's an area to watch, and watch your membranes and watch your buffer systems, watch your pH. Yes.
Yeah. Okay.
That's definitely an area to be very watchful for.
Yeah. And then, so just looking at time, maybe get a couple more questions in.
Yeah, sure.
So Shara is asking what could be the expected cost per dose for these vaccines versus established vaccines out there in the market?
Yeah, cost is always an interesting one. I have got this figure in my mind for the mRNA vaccines that they're probably in the order of something under £5 a dose. Significantly under, something around about £3 to £4 was, I think, a figure I've been quoted. Other figures for other vaccines that I think are out there. I think for COVID-19, I think the Chinese are charging around about $100 per dose. I think for some of the subunit vaccines we've heard mention of, certainly for when governments have been buying these things, of around about maybe $20 per dose.
Yeah, okay. And then Thomas is asking, is the plasmid manufacture off the critical path?
I guess it depends. It's if you're wanting pandemic response, the plasmid would definitely be on the critical path. There's all sorts of regulatory arguments as to, well, from the plasmid is only being used as a template, so does it need to be manufactured to GMP? And various people have different views on that. If it needs to be made to GMP, you would need to have establish a cell bank and have that tested and monitored, and that's not, I mean, it's E. coli you'd be dealing with, so it's not a hugely long process, but it's certainly take you a couple of months to do. So that's why I'd be quite interested in some of the non-plasmid approaches that using, for example, the touchlight technology to generate a DNA template that doesn't require the use of a plasmid and a GMP cell bank.
Yeah, okay. And then Sudhir, maybe we can come to you for your question, because I'm struggling to read red writing on a yellow background, so I can't really read the question. Hello, Sudhir?
Yeah, I can't really read it either.
Hello. Yeah, can you hear me?
Hi. Yes, we can do. Yes.
Yes. So I think, I don't know why it went like that. So I think my question was more on the mRNAs. They have an inherent adjuvant properties due to their complex interaction with the pattern recognition receptors. So does it warrant use of modified nucleotides? Do you see that is something which others are exploring, or for the vaccines, the traditional nucleotides will work?
Yeah. I should have mentioned that on the way through. The way that saRNA vaccines get around the limit of, I guess, the half-life of RNA in vivo is to just make more of it. The mRNA vaccines like Moderna or CureVac have been working on, they've typically been increasing the half-life of the mRNA by using a different set of nucleotides. And the big thing they've been doing is to use pseudouridine rather than uridine and you just shove pseudouridine into the transcription mix. And pseudouridine increases the half-life of the mRNA in vivo. In terms of adjuvanting, at the moment, the saRNA vaccines are considered to be self-adjuvanting. You're making this replicase, and the body is designed to recognize RNases as well through the capping system, and that's considered to be adequately self-adjuvanting. So I don't think any of the RNAs I'm aware of have any additional adjuvanting, but could be something for the future.
Thanks. Thanks a lot.
Hi, Andrew, you're still on mute.
Sorry, thank you. I was just saying, John, I don't know whether you can quickly answer Nenny's question in the interest of time. So in the context of clinical trials, the number of conducted studies using saRNA are relatively modest compared to the adenoviral vector. What are the strategies to expedite the development of the vaccine into the market?
Yeah, so I think that's the issue. There's of all the vaccines that have been looked at for COVID-19, the RNA vaccines are the ones that have had the least amount of data to support them, because they've just been newer. The adenovirus vaccines have had the advantage that they got very close to being used for MERS and SARS. It's yes, the RNA vaccines came along later. There is good animal data. There's a lot of work done by, a lot of the saRNA technology was developed by Novartis. If you look for, and they published a lot on that. A guy called Andy Gill did direct the team there. They did a lot of work. They had a lot of BARDA funding as well. They got very close to having taking an influenza vaccine into human trials. But then Novartis vaccines were sold to GSK, and I'm not quite sure how much GSK have brought this forward. And I think it's largely I think just the lack of experience. The regulators are very keen. I think it's just to get enough clinical data to support the efficacy of say the saRNA vaccines. But we should have data, I think, from end of November, early December on the efficacy.
Great. Well, I think as we're at time and there are no more questions, I'll just say thank you very much for a very interesting talk and discussion.
Okay, thank you all for your attention and your questions.